Okay, just imagine this for a second. What if you had the complete instruction manual for every single living thing on Earth? I'm talking from the tiniest virus to the biggest whale. Well, in just the last few decades, we've gotten incredibly close to that, sequencing thousands and thousands of genomes. But here's the catch, right? Having all the books is one thing, but actually understanding what they say, that's the real challenge. So how in the world do you find the secrets hidden inside this massive, massive library of life? Well, that's exactly what we're going to dive into.
And it's kind of hard to wrap your head around just how fast all this has happened. I mean, think about it. Back in 1995, we sequenced the very first bacterial genomes. You can think of those as like tiny little genetic pamphlets. Then, boom, by 2003, we'd finished the massive Human Genome Project. And today? Oh man, today we have a literal mountain of data. We've gone from a handful of pamphlets to a global library, and that's created this huge, incredible challenge. How do we actually read all of these books? All right, so here's our game plan for this explainer. First, we're going to look at the sheer scale of this library of life. Then we'll get into how scientists actually compare the blueprints inside. We'll check out the digital tools they're using, some of the big challenges they run into, and finally, why this all matters for, decoding our own story.
Okay, first things first. Let's just try to get a handle on the size of this library. Because when I say it's big, I mean, it's just mind-bogglingly, staggeringly huge. I mean, just look at this number. Over 2,200. We now have the complete genetic blueprints for more than 2,200 different organisms. And they're all just out there in public databases. We're talking everything from the simplest bacteria all the way to the most complex plants and animals, not to mention thousands of viruses. And every single one is a complete, unique instruction manual for a form of life. That's just incredible. So that brings up a huge question, right? How do you find the most important sentences in a library with thousands of books, especially when they're all written in a language you can barely understand? Well, the answer is actually pretty simple. You compare them. Let's dive into how that actually works. So this is the whole idea behind something called comparative genomics. It's really just the science of lining up the genetic blueprints of different species to see what's the same, what's different, and most importantly, why. It's kind of like comparing the architectural plans for, say, a regular house and a giant skyscraper. The basic principles of structure, foundations, walls, a roof, they're there in both. And finding those common, shared elements tells you what's absolutely fundamentally important. And here's what that looks like in action. When scientists find a genetic sequence, like this one right here, that's almost identical in both humans and, say, a mouse, well, that's a massive clue. It means evolution has protected that exact sequence for millions and millions of years. And that tells scientists one really important thing. This little piece of code must be doing something absolutely essential. Now, obviously, you can't just do this with a pen and paper. No way. This is a job for some seriously powerful computers and some brilliant software. We're talking about the digital tools that turn that massive data mountain into a map you can actually navigate. So the first question that probably pops into your head is, where do these genetic explorers even get their maps? It's not like they're just scattered all over the internet. Nope, they're kept in these huge, really well-organized digital libraries.
Yeah, scientists go to these massive public databases, and you should really think of them as digital libraries for genetics. These platforms don't just, store the raw code. They organize it. They create these amazing catalogs that can link a specific gene to a disease, or group proteins together into family trees. They even keep track of basically every sequencing project happening anywhere on the planet.
Okay, so we've got our libraries. They're full of books. But what kind of software do you use to actually read and compare the text across all of them at the same time? Well, this is where you need some incredibly powerful software. You can think of these tools as these search engines of the genomics world. They're these super complex algorithms that can take two huge genetic sequences and, just like that, find every single similarity and difference. We're talking everything from a tiny one-letter typo to whole chapters being moved around. So let's break down how a tool like this actually works. It's pretty cool. You basically feed it two complete genomes. The algorithm then gets to work, lining them up and comparing them letter by letter by letter. And what you get out the other end is this super detailed map that shows you everything. All the parts that match up exactly, any big repeated sections, and even those tiny single-letter differences, which scientists call SNPs. It's basically like getting a complete structural survey of two different blueprints. But, and there's always a but, right? This whole process is far from perfect. Like any exploration into a new frontier, there are going to be dangers and pitfalls. Just because you have a map, it doesn't mean you're not going to get lost. Okay, so here's a major problem. These automated tools, they learn from genomes that have already been studied and labeled by humans. It's a process called annotation. But what happens if one of those original labels was wrong? I mean, if just one scientist, way back when, made a tiny mistake identifying a gene's function, that single error can then get automatically copied and pasted across thousands of other genomes. It just amplifies the mistake over and over again.
And yeah, the algorithms themselves aren't perfect. Sometimes they'll flag a similarity that's really just a coincidence, that's a false positive. But what's even worse is when the settings aren't tuned just right and they completely miss a subtle but super important connection between two genes. That's a false negative. For researchers, it's this constant, tricky balancing act.
And this might be the biggest challenge of all. See, a gene doesn't just exist in a vacuum. The exact same genetic sequence can do totally different things in different organisms, all because the biological context is different. If you don't understand the unique biology of the specific creature you're studying, the raw data can be super, super misleading. Context is everything. So, with all these challenges, you might be asking, why even bother? Why do we do it? Well, it's because the view from the top of that data mountain is absolutely spectacular. The discoveries we're making by comparing all these genomes, they are literally transforming our understanding of life itself. And this is the payoff. This is what it's all about. By comparing our genome to other species, we can actually trace our own evolutionary history. We can finally start to figure out what thousands of these mysterious genes actually do. And maybe most importantly, we can pinpoint the precise genetic roots of human diseases. A great example, by comparing human DNA to mouse DNA, scientists were able to find the gene responsible for cystic fibrosis. That was a huge breakthrough that opened the door for developing new life-saving treatments. This quote really just nails it. It sums the whole thing up perfectly. This field isn't just about big, powerful computers and fancy algorithms. The ultimate success here, it all comes down to a partnership between the technology and real human expertise. You have to have that deep understanding of biology to make any sense of what the machines are telling you. So for the first time in all of human history, we have the ability to read and compare the source code of life on this massive, massive scale. It's given us this incredible, unprecedented power to understand disease, to understand evolution, and to understand our own place in the natural world. So the only real question left is, what story will we choose to write next?